Bioinfocongress III Dr. Kevin Young Protein mühendisi için çok modlu derin öğrenme. Dr. Kevin Young, 2011 yılında Ohio State Üniversitesi'nde kimya mühendisliği alanında lisans eğitimini tamamlamıştır. Ardından Inglewood, Kaliforniya'daki bir lisede 3 yıl matematik ve fizik dersleri vermiştir. Öğretmenlik yaparken aynı zamanda kendini daha etkili bir eğitmen ve robotik koç olmak için geliştirmiştir. 2014-2018 yılları arasında Dr. Young, laboratuvarı protein mühendisliğinde birçok gelişmeye öncülük eden Dr. Francis Arnold'un gözetiminde Kaliforniya Teknoloji Enstitüsü Kimya Mühendisliği alanında doktorasını tamamlamıştır. 2019 yılında Generate Biomedicine'da makine öğrenim alanında araştırmacı olarak çalışmıştır. Dr. Young şu anda Microsoft Research New England'da generatif modeller üzerinde çalışırken aynı zamanda proteinler ve kimya için ön eğitim, protein mühendisliği için makine öğrenimi ve sinir ağlarında belirsizlik ölçümü yapan kıdemli bir araştırmacıdır.
Bioinfocongress III Dr. Kevin Young Multimodal Deep Learning for Protein Engineering. Dr. Kevin Young received his Bachelor of Science in Chemical Engineering at the Ohio State University in 2011. Afterwards, he taught math and physics for 3 years at a high school in Inglewood, California. In addition to teaching, he advanced in being an effective instructor and robotics coach during these years. Between 2014 and 2018, Dr. Kevin Young studied as a PhD student in chemical engineering at the California Institute of Technology, Caltech, under direction of Dr. Francis Arnold, whose lab pioneered many developments in protein engineering. He worked as a machine learning scientist at Generate Biomedicines in 2019. Currently, he is a senior researcher at Microsoft Research New England, working on generative models and pre-training for proteins and chemistry, machine learning for protein engineering, and uncertainty quantification in neural networks.
Dear Dr. Kevin Young and participants, welcome all of you to our Bioinfocongress III event. Dr. Young will give us a talk today on multimodal deep learning for protein engineering. At the end of the presentation, we will have a question-answer section prepared by our team and consisting of questions from our participants.
All right. So today I'm talking about multimodal deep learning for protein engineering.
Dear Dr. Young, welcome back. Can you hear me
Hello?
now?
Yes. I can hear you.
If you wish, you can start your presentation.
All right. So today I'll be talking about multimodal deep learning for protein engineering. So proteins are biology's actuators. Human cells contain between 1 and 3 billion proteins each. They're responsible for things like structure, metabolism, and signaling. For example, IgG is part of your adaptive immune system. It helps to recognize the proteins of pathogens so that your immune system can get rid of them. Hemoglobin carries oxygen in your blood. Insulin helps to regulate your blood sugar. The coronavirus spike protein helps COVID-19 virus, the SARS-CoV-2 virus, get into your cells. And luciferase is a fun one because it makes firefly butts glow. All of this functional diversity arises from 20 building blocks called amino acids. And they're called amino acids because they have an amine group on one end and a carboxyl group on the other. And then what makes them different from each other, the 20 amino acids have different, what are called side chains. Here we're using R. And for example, the simplest side chain is just a hydrogen for glycine. You also notice that every amino acid has its full name, a 3-letter code and a 1-letter code for notational efficiency. Next, we have alanine, which has a methyl group for its side chain. Serine has an a COH group. And then threonine has another methyl off that. And cysteine has a sulfur. And there's 20 of those of various sizes and charges and chemical properties and pHs. And all of this, from these 20 simple building blocks, we get all of that functional diversity I talked about earlier. So why might we want to design proteins or engineer proteins? One reason is to get new chemistry. So a few years back, people engineered people engineered enzymes to make carbon silicon bonds, which is a reaction that people had done with heavy metal catalysts before, but is not known to be done in nature by enzymes. But turns out proteins can do it. You might want to understand the function. So this is AAV, which is a virus that can integrate bits of DNA into host cell genomes, and it's of interest for genetic engineering and gene therapy. We might use the protein itself as a therapeutic. So, for example, this is asparaginase, which breaks down the amino acid asparagine. Many tumor cells cannot make their own asparagine. So if you break down all the asparagine in the environment, the tumor cells die, and your healthy cells, which can produce their own asparagine, go on happily. And we also use proteins as molecular tools, the most famous of which is green fluorescent protein, or GFP, shown here. And you can use this to tag parts of cells and then visualize them in your microscope in visible light. To help us engineer proteins, we have access to very large sequence and structure databases, including BFD, with about 2.5 billion sequences. If you want a deduplicated database, there's URF100 with 314 million sequences. If you don't want to overrepresent common proteins, you can cluster them down to 90% or to 50%. If you only want experimentally verified proteins, there's SwissProt with 568,000 experimentally verified protein sequences. If you want structures, you can look at the PDB, which has 194,000 sequences and structures. And compare this to a typical directed evolution campaign where you might make a few hundred measurements. So there's a lot of data out there that we can try to bring in. And so a big part of my research agenda is to use multiple data modalities from these databases to discover and design proteins. So the general paradigm is we want to train an initial model on natural protein data, and that can mean sequences, annotations, structures, perhaps some reaction data, free text, some energetics, and combine some subset of those into one model, and then we can use that model in two ways. One of those ways is as a pre-trained protein fitness predictor, and the other is to generate de novo proteins with desired function. So right now we're going to focus on pre-training, and we're actually going to look at not just individual proteins, but clusters of proteins. So there are these things in bacteria called biosynthetic gene clusters, and the encoder are known as natural products. So in your microbial genome, many microbes have these co-located and co-regulated genes. That's why they're called gene clusters. They tend to modularly produce secondary metabolites. So secondary metabolites are responsible for things like toxins to clear space for itself, or signaling with other microbes in the area. And as a result, many of these are very useful for, here's an example of one. You can see on top, we're looking at the genome. We're looking at some genes on the left, and then these two big genes in the middle, and then some more genes on the right. And when I say these are modular, not every class of biosynthetic gene clusters work this way, but many of them have these chunks of the genome encodes proteins that each work one step at a time to build some molecule. And in this case, it builds thermoactinoamide A. This is very big, kind of scary-looking molecule. And people are interested in these as sources of chemicals in general, but also especially of therapeutics and pharmaceuticals. Some examples: penicillin is a natural product made by fungi. Vancomycin is a more modern antibiotic. It's also based on a natural product. There's a lot of cancer drugs that are derived from natural products, like dipiracil and belinostat. And here, darunavir is an anti-HIV drug, and voglibose is a diabetes drug. So this is some examples of drugs derived from natural products or based on synthetic versions of natural products. Unfortunately, BGCs are somewhat difficult to discover and characterize. So you have these microbial genomes, and so you have to figure out which genes are part of a cluster, what are the boundaries, and what does it make? And to make this extra challenging, many of these microbes, we have their genomes. They live in the sea or in soil somewhere, and we can't culture them in the lab. And it's pretty difficult, even if you can get metabolites from a sample, to map that to genes, or if you have the gene, to map that to the product. And how difficult is this? To date, there's a database called MIBIG, which is Minimal Information About Biosynthetic Gene Clusters. It only includes gene clusters that are well characterized. I mean, we know what genes are in there, we know what it makes. And there's only about 1,400 in that database. So we're going to turn to some methods that pretend proteins are language and use that to see if we can do some unsupervised learning here. So there's a lot of models out there that treat amino acids like tokens and use tools originally developed for language, such as word2vec, where you make what's called the distributional hypothesis, that similar words with similar meanings appear in similar contexts. So here we're saying amino acids or small chunks of amino acids with similar meanings appear in similar contexts. And you can use that to get embedding, but you can use to train downstream models. And then people came along with neural networks and said, we can do an autoregressive language model, meaning you predict the next amino acid from the previous one. And you can use that second to last layer to represent the protein, or each position of the protein. And then people came along for that and said the problem with this autoregressive model is that at every location, we only look backwards. We would like to look in both directions. So to do that, we're going to corrupt a sequence by masking or changing certain amino acids, then pass it through a neural network and predict the original uncorrupted sequence. And once again, you can use that embedding for downstream tasks. But today we're not going to talk about, right now we're not going to talk about individual proteins, but back to these BGCs. So we trained a model called BigCarp, which stands for Biosynthetic Gene Cluster Convolutional Autoencoding Representations of Protein. So before this, there was a model called CARP, which was just on the amino acid level. So now we're going to do this at the BGC level. To do this, we're going to carry the data set. We're going to possibly use some pre-trained amino acid level embeddings. We're going to train our model, our self-supervised model. And then we're going to use it to predict biosynthetic gene cluster product classes. So what kind of natural product does it make? or we're going to use it to detect BGCs in bacterial genomes. And this was an internship project with Carolina Rios-Martinez and Nick Bhattacharya, who were both at Berkeley before their internship. And I think Nick has graduated now. Carolina is at Stanford in grad school. So previous data mining efforts use anything from a linear model to a hidden Markov model to an LSTM to find positive examples of BGCs. And true negative examples are pretty hard to find because maybe it's just part of a BGC we don't know about or a kind of BGC we don't know about. So first, curate data set, starting from microbial genomes. We go and find all the open reading frames. And then we use a tool called anti-smash, which is based on the Markov models to identify what it thinks BGCs are. And from this, we get 127,000 BGCs with 55 different product classes. And now we're going to do something kind of new here. So people have trained these mass language models on language, obviously, but also on amino acid sequences, on DNA sequences and RNA sequences, on glycans. But we're going to do this on the domain level. And the domain is a protein subunit with compact structure and modular function. So for example, the cytochrome P450 from the BM3 has 3 domains. It has an FAD domain and an FMN domain. And both of those help with an electron transport chain that grabs electrons from an ADPH and transfers that to the P450 heme domain, which is the actual catalytic domain. But each of those domains folds by itself as a compact structure and kind of does its own thing, and they pass electrons to each other in this case. And so we're representing things as at the domain level as a compromise between how fine-grained our representation is, but also how big the vocab size is and how long the sequences we're going to have to deal with are. Some examples of domains you might find in BGCs, and the PF stands for PFAM. So PFAM is a database that tries to catalog all the protein domains out there. PF stands for PFAM, kind of stands for protein family, but not officially. So some examples you might find in our data set are glycolic acid cyclopropane synthetase, which is an enzyme, or ketoreductase, which is also an enzyme. So we've got these BGCs we've identified, and we can take a look at these BGCs and identify in the open reading frames different PFAM domains, which we're representing with those colored dots. And we're just going to pull those out and represent the BGC as a sequence of domains. And so we have 127,000 BGCs, 19.5 unique domains, and 55 product classes. So now we're going to do this mass language model self-training, where we train BigCarp to reconstruct corrupted biosynthetic gene clusters in the domain level. So we have our self-supervised data set. To each one, we're actually going to prepend a token that represents the product class, which also gets learned along with the sequence. We're going to mask and corrupt some of the tokens, about 15% at every pass, and train the model to reconstruct the original PFAM domains, or if the product class token gets masked, to reconstruct the original product class. So in this way, you implicitly train a model that learns what kinds of domains and in what order belong in which product class, but also can predict the product class from a sequence of domains. And zooming in a little bit on what BigCarp is, there's an input embedding. So we embed the PFAMs into a 1,280 dimensional space, then embed that into a d-dimensional space. D for these is 256. And then we have what are called bytenet blocks, and then we do an output layer norm feed forward and softmax to get output probabilities. And each bytenet block has layer norms, some 1x1s, and what's really important in the bytenet block is that 1x3 dilated convolution, which is what allows the model to look at the entire sequence as you go through the model. And this model has about 34 million parameters. And we're also going to examine that PFAM embedding a little bit more. So every PFAM domain is made up of a canonical sequence of amino acids. So there are, as I said earlier, people have trained these mass language models that take in amino acids. So if we just look at one domain at a time, that's a sequence of amino acids, you can run that through a pre-trained model. In this case, we use ESM1B, and use that to represent that particular PFAM in the input of BigCarp. And so we try three things. We try using ESM1B and then as an initialization for our PFAM embeddings, but then training them along with the rest of the model. We try using ESM1B to initialize the PFAM embeddings and not letting them train with the rest of the model, or randomly initializing those PFAM embeddings. And all three versions of the model manage to reconstruct BGC domain sequences. So on the left, you see the loss by epoch. They all go down. And these are all validation curves. And on the right, you see accuracy by epoch. They all go up. And the frozen one seems to do a little bit worse on validation here than either the random or fine-tuned. But the gap isn't that big. And if we look at the pre-training test set on a domain level, so we're looking at exponential across entropy. So lower is better. Lower means it's more confident about the correct domain every position. And so this correlates with what we see in the plots. The fine-tuned and the random models for the PFAM embeddings do a little bit better here. But we also look at the product classes separately. So once again, lower is better. And here we find that actually the frozen model is a little better. But all of them learn. And if we look at those input embeddings, we actually find that they encode some relevant PFAM information. So what we're going to look at is a two-dimensional UMAP. So, and we're only going to look at 10 PFAM clans, just because otherwise there would be a zillion colors and no one would be able to make any sense for them. These are the most common PFAM clans in our data set. So I'll sort by Does that work?
Yes, Hocam, we can hear you.
Okay. Let me. How's that? Sorry, my headphones just turned off. Just unfortunate. How's that?
Yes, sir. We can hear you. You can continue the presentation.
Okay, cool. Sorry. So here we're going to look at the frozen embeddings, which are just what you would get from ESM1B without any further training. And here we can see that the PFAM clans cluster in the embedding space, which makes sense because the PFAM clans have different sequences. And we can see that in the fine-tuned ones, even after fine-tuning for BGC prediction, the domains are still pretty clustered. And interestingly, the random ones don't cluster, but they still manage to learn the domain reconstruction task, which is interesting. Okay, so that was pre-training or self-supervised learning. Now we want to see if we can do anything useful downstream with these. So first, we came up with kind of a clever method for doing unsupervised BGC detection with this model. And so we have a set of 9 bacterial genomes that were not in the training set. And we're going to consider all windows of 64 PFAM domains. We're going to append a mask token to the beginning and run that through the model. So here my goal is to predict whether any given PFAM domain is the first domain in a cluster. And the way I'm going to do that is by looking at how certain the model is of what that mask should be. So if this is the real start of a PFAM of a biosynthetic gene cluster, it will look like something the model has seen before, and the model should be very certain about what the product class should be. Because remember, during pre-training, we always start with a product class. So the more certain the model is about what product classes should be, the more likely it is that this is the true beginning of a BGC cluster. And we're going to do this at every location in these 9 genomes and look at the area under the receiver operating curve for start locations. And all 3 of these actually do pretty well. And interestingly, the best one is the random model, even though those embeddings look not as great. We also want to be able to predict whether any individual domain is part of a BGC. And the way we're going to do that is using the same data, same predictions. We're going to look at every time a domain is predicted, how certain the model is that the original domain was not corrupted. And the idea being here if this domain is part of a BGC, it should look like something the model has seen before. So it should be pretty certain that it's uncorrupted and belongs there. So same thing, we're going to look at the area under the receiver operating curve for every PFAM domain in these 9 bacterial genomes. And here the ESM1B fine-tune does the best. And we see that Frozen actually underperforms on both of these tasks. So probably being able to learn those PFAM embeddings with the pre-training task is pretty important for these tasks at least. Following some previous work, we also tried to see what would happen if we fine-tune with negative examples. So we took those 1,406 BGCs from MIBIG. These are the very well characterized ones, which are also not in the training set. And 10,128 synthetically constructed negative clusters and run them through BigCarp. And for every domain, predict probability that it's in a BGC. So we're going to look at the 3 pre-trained models, the same architecture without pre-training, and DeepBGC, which is an LSTM. It's a supervised LSTM, no pre-training. And this is the same data set they train on. So in validation, the fine-tune does best, but all of our models do better than DeepBGC, even without pre-training. So our architecture might just be a little more useful here. And on test, fine-tune still does very well, but so does the no pre-train case. Well, all of these are very close. So in general, I would say that once you do add negative examples, the pre-training doesn't give you a huge boost. And then we also want to predict BGC product classes. So the way we do that is we're going to test on those same 1,406 BGCs from MIBIG. And we're going to take these real BGCs, append a mask token in the beginning, and run that through, and just take the class prediction. So MIBIG has these 7 product classes. We're going to ensemble the predictions from our 3 pre-trained models and compare to DeepBGC, which sticks a random forest on top of their LSTM. And they actually do report cross-validation results on these 1,406 BGCs, whereas our results are not trained on this data set. They're only trained on the pre-training data set. So on average, we do a fair amount better. This is, once again, area under the receiver operating curve. And per class, for 3 of the classes, we do better. For 4 of the classes, we do better. For the other three, they do better. But the ones where we do better, we tend to do a lot better. All right, so to summarize this part of the talk, BigCarp detects and classifies PGCs. Here's our pipeline. We created a data set. We tried some different pre-trained PFAM embeddings. We train our self-supervised model. And then we can predict product classes, and we can try to detect PGCs. As far as we know, this is the first masked language model trained on protein domains and trained on PGCs. And we can do unsupervised PGC detection, and we learn the product classes and the PGC structure simultaneously. So that's the first part. So back to this kind of overview slide. Now we're going to consider a case where we're going to focus mostly on structure, and we're going to try to generate new proteins. So the information flow in protein function goes from sequence to amino acid sequence, which folds into a 3D structure, and that 3D structure determines its function. If we can generate new designable structures, designable meaning we can find an amino acid that will fold to that structure, that will expand the function space we have access to. So for this part of the talk, we're going to focus on generating new structures with the hypothesis and the expectation that they'll lead to new functions. And the challenge, as always with generative models, is to generate diverse and realistic or designable things. This was also an internship project by Kevin Wu, who was a PhD student at Stanford. And we're going to use something called a diffusion model. And these diffusion denoising probabilistic models learn to generate objects from noise. So they were first demonstrated in images, the idea being that you could take a 128 or 1 by 128 patch of Gaussian noise and run it through a neural net and get back a realistic looking face. How does that work? Well, during training, you start with the faces and you go from, which is x0, the original uncorrupted object, and you go take time steps, and every time step you add a little bit of noise until at time t big, it's indistinguishable from a sample from a Gaussian, an isotropic Gaussian. And during training, what you do is you take a noise example, x little t, run it through a neural net, and try to predict the previous step, or to predict the noise that was added to the previous step to get the current step. And then at inference, you can start from noise and just run the model over and over and over again and get back examples of images. Of course, we don't care about images. People can draw, people can take pictures. It's much more interesting, in my opinion, to make machines do things that people can't. And maybe there's a few people in the world who can dream up new, diverse, complex protein backbones. I am not one of them. And we're going to generate what are called backbone structures. So if we look at this amino acid, general amino acid, there's an amine, there's a carboxylic group, carboxylic acid group, and there's a side chain. And the side chain, remember, is what makes the amino acids different from each other. And the backbone means we're only going to look at the non-side chain atoms. And what that lets us do is not specify what amino acid is at which position while we're designing the overall structure. And this carbon here is generally called a C alpha carbon. And there's some previous work using diffusion models to generate structures where they just generate the locations of the C alpha carbons and fill in everything else later. And they do this as a point cloud. So here's an example, and this is taken from Namrata Anand's website and from their paper, where they start with these random locations or Gaussian locations for the C alpha carbons, and then diffuse them and then fill in the rest of the protein around them. To do this, your neural network must be equivariant to translation and rotation, because two structures are the same, even if you shift them left or right or up or down, or if you rotate them around. Models trained like this for proteins, the previous work, tends to generate a lot of left-handed helices instead of right-handed helices. So in nature, you see some left-handed helices, but not very many. So we thought carefully about this. By we, I mean mostly Kevin. We thought carefully about this. And the other problem with this point cloud 3D coordinate diffusion is that it kind of ignores any biological intuition. So we thought about how a protein actually folds, which is gets synthesized in the ribosome, and then it kind of wiggles around. And there's only certain parts of it that can wiggle, right? It can't fly apart. The adjacent amino acids that are covalently bonded, it can't be on the opposite sides of the world. There's only certain degrees of freedom. And the degrees of freedom we're going to consider are these 3 bond angles, thetas 1, 2, and 3 in green, and these 3 torsional or dihedral angles, phi, phi, and psi, in yellow. And these exist for every pair of amino acids. So here you can see there's an R1 and R2. This is 2 amino acids. There's 2 C alphas. You can define these 6 angles. And if you define these 6 angles and the C alpha distances for every consecutive pair of amino acids, you can fully specify the structure. We're only going to consider the angles because that covalent bond distance doesn't vary very much within the structure. And by using these coordinates, we can throw out the whole complex model thing and our model folding diffusion. So we use these coordinates inspired by protein folding. And so at x0, we have the real coordinates. And then we just add simple Gaussian noise with a wraparound so that we can't go past 2 pi. We also center the angles. So that xt big, we have random angles with a funny looking noodle structure. And then x0 is a nice looking structure, in this case with a lot of helices. And then our model, we use P, our model P predicts at every step the noise added to, and then we can go backwards from random angles to real angles. And we do this with a vanilla transformer. So there's no fancy equivariant models. We just took a transformer from Hugging Face. And we trained this on 30,000 structures from CAF. So CAF is deduplicated, so they're diverse structures. And we split them randomly, 80% train, 10% each validation and test. And we also only consider structures that are at least 40 residues. Anything longer than 128, we crop randomly during training. So first, let's look at the angle level. There are 6 angles. Remember what they are. Here I'm showing their cumulative distribution, cumulative frequencies between the test set and what we sample. And the takeaway here is that these look very similar for all 6 angles. So if you consider each angle independently, the distributions we learn match. Of course, this is not enough to generate good proteins. So let's look at pairwise distributions. So the historical way to look at these is to look at the plot psi and phi together. And the reason is that your location in this plot, which is called a Ramachandran plot, they can tell you about what secondary structure you're in. So up in the upper left, you can see that's where beta sheets are. And then right-hand alpha helices are kind of in the middle lower left. And you can see the left-hand alpha helices are over on the right side. And we're just looking at the frequency of, this is the frequency plot of how often each of these pairs of angles co-occur. And so the left is the real distribution, and the right is our generated distribution. And you can see they look pretty similar, which is great. We can also actually compute the secondary structures that we generate. Mostly, we can consider where the alpha helices are, where the beta sheets are, and see how often we're going to count the number of alpha helices and beta sheets in your structure and see how often they co-occur. So this is the test distribution and on the right is the generated distribution. So ours is a little sharper than the test distribution, but they look pretty similar. And if we just inspect the structures manually, they look pretty reasonable. Lots of helices. We wish we could see more sheets, beta sheets, but they do occur. Of course, once again, our ability to visually inspect generated backbone structures and see if they're reasonable is pretty limited unless you're one of a few very, very talented people. So one disadvantage of working with generative models of proteins as opposed to images is that you can't get easy by eye evaluations. So recently there's people have been measuring what's called designability of these structures with a metric called self-consistency TM score. The way that works is first you take your model, you generate a bunch of things, you take each of those and run them through a model that predicts the amino acid given the backbone structure. And in this case, we use ESMIF. Then you run that through a model like AlphaFold, or in this case, OmegaFold, that predicts the structure given the amino acid sequence. And what we would like is for those structures to be similar or identical, actually. So TM score is a metric that ranges from 0 to 1. 0 is these are not alike. 1 is these are exactly the same. And so we're going to calculate the TM score between the generated structure and the OmegaFold structure and call that the self-consistency TM score. So high means that ESM and OmegaFold are able to generate a sequence that folds exactly to the structure. 0 means we couldn't get anything that folds anywhere close to it. Generally, people consider anything a TM score above 0.5 to mean they have the same fold. So that's kind of the cutoff people look at. And many of our generative structures are designable. So here we're looking at, we've divided the generative structures into those with fewer than 70 amino acids, so 40 to 69, sorry, 40 to 70. And then the longer ones are 71 and 128. In general, the short ones, there's a lot above 0.5, and there's a few long ones above 0.5, but those seem to be a little harder. So we compare this to a previous work called ProTIF by Brian Tripp, who also interned with us last year, but did not work on this with us. And for generated samples smaller at less than or equal to 70 amino acids, we do a lot better than they do, 57 as opposed to 36 out of 210. Greater than 70 amino acids, neither of the models were great, at 54 and 51 out of 570. So less than 10%. And then, so we also want to consider some baselines. So the simple one here is if I just take the sets of angles in my train set and shuffle them and sample that way, that preserves the Ramachandran plots, but it doesn't preserve longer correlations. And what does that look like? Well, the self-consistency curves, self-consistency TM scores there are very bad. So the randomly generated samples in blue, I don't think there are any above 0.5. And you can see if we generate from folding diff, you can get about 11% above 0.5. So we do beat the random baseline, that's good. We also want to make sure our structures are diverse. So the top row here is generated structures. The middle row we're showing in our training set the most similar structure by TM score. And those for these 4 that we picked range from 0.6 to 0.71. So not identical. And many of them are pretty dissimilar. And then the bottom row is, if we run this through ESM inverse folding and get an amino acid sequence and run that through OmegaFold, what structure do you get back? And then we also report the self-consistency TM score. So higher is better there. The ones we picked here aren't great. We only picked one that's above 0.5, but you can see in many cases, they still have at least a passing resemblance. But the big takeaway here is that we're not generating things that are exactly like the training set. We can look at this another way. We can take all 580 of our things and cluster them by TM score. You can see you can kind of pick out a few clusters, but the clusters are not huge and they're not super well defined. And we contrast this with ProDiff, which is a prior diffusion model on just the C alpha carbons. And you can see that they have 11 very well-defined blocks of structures in their 780 generated samples that they report in their paper. We can also look at, we can also just plot for all of our samples the max training TM score and the self-consistency TM score. So the goal is to generate things that are very self-consistent and very dissimilar from the training set. And those are very hard to get, unfortunately. But we do manage to generate things that are more similar to, have better self-consistency scores than their maximum training TM score. And of course, the way these work is that you are generally going to see a correlation between those. Finally, we want to check that our improvements come from our angle formulation and not just from transformers being magic or all you need, as some people claim. So to do that, we retrain the model using C alpha coordinates only, the way previous diffusion papers have. And here it's a little harder to look at Ramachandran plots or to look at angles in general, but we can still look at the C alpha distances. So here is a good example of what those C alpha distances should look like in the real protein. You can see there are parts that there are significant off-diagonal parts that are in close contact with each other. And ours also kind of looks like that. Ours looks maybe a little too idealized, would be my take here. A little too symmetrical. But if you just do a transformer on the coordinates, you basically get a squiggle where nothing is close to anything else. So a transformer without equivariance cannot work on point clouds, which might sound obvious, but we thought it would be thorough and take care of that. And of course, folding diff is the first step towards generating new functions. So as a reminder, we're doing diffusion on internal coordinates inspired by physical protein folding. We can generate realistic and diverse samples, although of course we want more things in that upper left-hand corner. And hopefully soon, either computationally or in the lab, we or someone else can use this to generate new and interesting protein functions. So this is out in preprint. The code is available on GitHub, and it's available on Hugging Face, depending on your preference. So go forth and generate new backbone structures. All right, so to summarize overall, my kind of big picture research interests are predicting and generating functional proteins or sets of proteins using multimodal data. And that data might include sequences, annotations, structures, chemical reaction information, free text, energetics. And we use that to train an initial model, and we can use that initial model to predict protein fitness or to generate or discover de novo proteins. And I acknowledged the students who worked in these projects on the project slides, but also want to acknowledge people at MSR New England. So this is the BioML group at MSR New England. And the projects here were done with Ava Soleimani, who's on the top left. Next to her is Alex Liu. And then at the top right is Lauren Crawford. So they worked on one or both of these projects with me, along with the interns. And with that, I'm ready to take questions.
Thank you very much, Dr. Yang. If you wish, we can go to the question and answer section prepared by our team and consisting of questions from our participants.
Yes, go ahead.
Okay. First question, can BGC-associated transporter genes inform predictions of specialized metabolite structure and function, including siderophores and antibiotics?
BGC-associated transporter genes inform.
If you wish, I can repeat the question. Sorry.
What's a BGC? I'm not sure what the BGC-associated transporter gene is.
Okay. If you wish, we can continue with the next question, and if we have time, we can go back this question later. Okay?
Yeah. Sorry. This is a
it's okay.
rabbit hole here.
Thanks. Next question. How is it possible that convolutional networks can be developed on localization of classification to learn protein representations? Are these approaches could be encapsulate diverse functional information and significantly outperform autoencoder-based models?
Right. So these convolutional models are essentially masked autoencoders, right? They're exactly the same as ESM or BERT, except that instead of using the transformer, they're using dilated convolutions. And on proteins, we find that using convolution across tasks does approximately as well as ESM as a transformer of the same size. The intuition in natural language is that this is true up to like 1 billion parameters, and then the transformers just scale better. Until recently, no one had trained a transformer that big, and for proteins, and I don't think anybody's definitely no one's trained a COMNET that big. So it's hard to know if that hypothesis holds true. So the advantage of a COMNET is that it scales linearly with sequence length and computation and memory. And that matters practically because there are certain proteins like CRISPR-Cas9, which are about 1,300 amino acids, which really strain the memory requirements of, say, ESM1B, which is a 650 million parameter transformer model. Right. The advantage of transformers for proteins, and ironically, even though they're developed for natural language, I would argue that transformers are maybe an even better match for protein modeling because those attention maps. It turns out that those pairwise interactions are really important in stabilizing protein structures. And before AlphaFold, people used coevolution and contacts as a structure prediction method, right? And even now, you can get pretty far by just inferring contacts from MSAs. And it turns out that if you use a transformer to do mass language modeling, it explicitly learns those in the attention maps.
Thank you for your answer, sir. I'm continuing with the next question. What opportunities does equivalent networks design in deep generative models inspired by the in vivo protein folding process that works on residue angles in protein backbones rather than Cartesian atom coordinates offer in protein structure generation?
Right. So right now, given how fast this field moves, this may not be true in the next week, but right now, folding diff for unconditional protein structure generation with diffusion gives you the best generated backbones. So what I would like to see happen in the future is, one, combine that with sequence generation, and two, you want to generate not just unconditional structures, but you want to condition on, here's the motif I want, and I want to generate around it, which Brian Tripp's paper actually addresses very thoroughly. And it's obvious next step is to combine our angle formulation with that and see if that works. But more ambitious things like recently, Google released a model that, given a protein sequence, will output text to describe the protein, and Unported is actually using it now. But can we go the other way? Can you generate some text? Can you pull in some text and generate a structure that matches that text? Would be really cool if someone wants to try that.
Thank you for the answer, sir. Next question is coming from our participants. The premise of the SSL technique is to not use labeled data. The downside to that approach is you either need huge amounts of data to generate accurate pseudo labels or compromise on accuracy. Does Big Carp generate inaccurate labels? How do you scale accuracy? Thank you.
Right. So we actually have a fair amount of unlabeled data, right? Because UNIREF 50, which is kind of the standard data set nowadays for sequence-based pre-training, has about 50 million sequences nowadays. If you don't care as much about data quality, you can use BFD, which is 2.5 billion sequences, which gets you in the range of like the big text databases. As for whether these models are accurate, it's hard to say on downstream tasks, right? So I gave a whole talk on this on Monday, but this mass language modeling task seems to do a really good job of teaching coevolution, teaching structure. And like OmegaFold, which you use here, uses a language model to implicitly get MSA, multiple sequence alignment information, to predict structure from single sequences. Where these pre-trained models don't do as well, unfortunately, is say I have a protein, I measure all the effects of single mutations, and I want to predict the effects of double and triple mutations. Here, that expensive pre-training doesn't give you the huge boost it does for structure prediction.
Okay. Thank you for the response. And again, this question is from our YouTube participants. Do you think that multimodal models increase the complexity and cost of data collection, making the consistency of algorithms more difficult and difficult to interpret?
Right. So depends what those modes are, right? For a lot of these, for example, sequence and structure, nowadays you just pull them from the database, and they're public databases. If you want, say, annotations, like people already have pipelines to do that, right? All the modes, most of the modes I've talked about there, people already deposit those in the databases. Maybe not as thoroughly as they deposit sequences, but you can find a lot of them. The exception on my slide is energetics. Those you have to find to decide how you want to compute, and you have to generally have to compute them yourself, which takes some expertise. As for interpretability, so the difficulty is that if the possible downside here is if your model takes in as like input sequence, and you need annotations, and you need text, and you need structure as input, that might be difficult to provide at inference time for new proteins, right? So you have to kind of find a way to store that information and not need it at inference time, ideally. And as for interpretability, that once again depends a lot on what kind of data you're using. So if you're going straight from sequence to like a mutation effect prediction, that can often, with a neural net, that's going to be hard to interpret, because if you're doing it, if you have a neural net that does this perfectly, it's implicitly learning energetics and structure, but it can't tell you any of that, right? Now, if you take your network and make it explicitly use energetics and structure, then maybe it's interpretable. But depending on how you use it, maybe not. So I think that depends a lot on the specific data modeling choices. And it's hard to make a big call on interpretability there. There's also a part of me that always wants to say that if there were an easily interpretable way to make these predictions, we'd have found it already. And we're using neural nets because there isn't. But that's a debatable point. That's just mostly my opinion.
Thank you for the answer, sir. Next question. What important steps does your research on protein structure generation by folding diffusion point to in developing new proteins and protein-based therapeutics?
All right, I think we're still, the structure generation part is still kind of at the basic research stage. We're trying to show that it's possible, and we're trying to come up with creative ways to do it. And we don't really have a good idea what the best method is or what the most useful method is yet. And really, like I said earlier, to be useful, you have to be able to specify the function you want, right? Because no pharma company wants to just like generate random scaffolds to test things on. And no enzyme companies don't want to do that either. If I wanted to have random scaffolds to test, I'm just going to go take a metagenome sample from dirt somewhere. I don't necessarily need a neural net to do that.
Thank you for the answer, sir. Next question is coming from our participant. Firstly, thank you, sir. Are there any points to be considered in biological data training models when compared to common data training models?
This one's a little hard to answer because I've actually never trained models on non-biological data. But one big difference in general models, I touched on this earlier, is that it's hard to evaluate samples, right? Because in images and text, you can look at them or read them and have some idea of how good an individual sample is. And then you can come up with creative ways to like evaluate sets of samples. But like I said, I can tell that the protein has some helices and sheets. Beyond that, it's hard to say if the structure is good or bad. If you start generating the sequences, it gets even harder, right? I can see it starts with an M. It doesn't have a ton of Ys, which is probably good. And beyond that, it's kind of hard to say. A nice thing is that at the molecular level, at least, is that there are a lot fewer societal and ethical concerns than when you're scraping the internet for images or text, because then there's IP issues, there's ethical concerns of like what kinds of things are you automating or generating, what kind of biases are you perpetuating, which at the molecular level in these public data sets is much less of a concern. Now, of course, once you start moving up a level and doing human subjects and health, then it becomes a huge concern. But yeah. But otherwise, most of my knowledge of how people do machine learning on things that aren't biology is observational. So.
Okay, thank you for the answer, sir. Next question. What robust evaluation strategy can be used to assess quality of protein representations across different scales of biological function?
So I don't think there is one evaluation strategy to assess your representations. It depends on what your downstream goal is. And at the moment, it doesn't look likely, unless we solve physics in the next couple of years, that there will be one representation that will be the best for every goal. Just like there's not one model that's best for every problem.
Okay, thank you for the answer, sir. Next question. For example, AlphaFold 2 requires computational resources equivalent to 100 to 200 GPUs running for several weeks. Which methods do you think can be applied to reduce memory and computational costs in data training?
Right. So the 100 GPUs for several weeks is training on inference, right? Because you can run AlphaFold on one GPU for inference. Training is hard because you have to try different things. And so they report like the final model was trained with 100 of the couple hundred GPUs for a couple weeks, but all the models they tried before that didn't work as well. And all the ablations they did also each took 100 GPUs for a few weeks, and that multiplies really quickly. So how to do better, one way, the kind of high-tech way, is these kind of AutoML methods, right? That use statistical methods. They'll often use Gaussian processes to say, given this set of hyperparameters, given how training curves usually look, you should run this set of models for this long, and then these are promising to run this longer, maybe scale them up, that kind of thing, right? Unfortunately, that takes a lot of technical sophistication to have that kind of setup. Other things are just coming up with creative, non-brute force ways to model things, right? So the final AlphaFold architecture is actually fairly creative and not just like a brute force thing. So what takes a long time in training is the recycles and then the data augmentation. But I don't know if there's really a good answer there beyond be creative.
Thank you for the answer, sir. And a question is coming from our participant. Data scarcity, it's one of the most challenging problems in growing a deep neural network using medical data. A state-of-the-art approach to deal with the lack of data is few-shot learning. Should few-shot learning be more widely researched in multimodal medical data analysis?
So honestly, I don't know that much about medical data. I usually do molecular things. But in protein engineering, a lot of the things we want to do, you could frame them as few-shot learning, right? Or zero-shot learning. I want to know the effects of these mutations on this thing I want to measure. And I want to take as few measurements as possible. So that, or even no measurements for zero-shot. But yeah, I don't really know about medical data.
Okay, thank you for the answer, sir. Next question, again, a participant. Thank you very much for your informative presentation. I would love to hear your thoughts on combining machine learning models with evolutionary algorithms for protein design. Best regards.
Right, so depending on what you mean by evolutionary algorithms, one way obviously is machine learning guided direct evolution, where you train a small data set and you use a machine learning model to predict what variants you want to measure next. And I think that's been pretty well proven in the last decade or so, even though it actually started way back in like a few decades ago with people using just linear models even. And generally using any model, even a linear model or something very simple, will do better than randomly choosing things. We could also use this in the context of in silico kind of direct evolution, right? So if you have a machine learning model you think is very good, you can treat it as an oracle. You can try to use an evolutionary algorithm to optimize sequences according to your oracle and hope they work in the lab. The difficulty there is that big assumption that your neural net is an oracle and that you won't just find adversarial examples.
Okay.
Sorry, I hope that answers the question.
Thank you for the answer, sir. Next question. Do multimodal deep learning systems contain multimodal specific challenges, such as missing all modalities? What is being done to make protein engineering more clinically relevant?
More what? Sorry, what's the end?
What is being done to make protein engineering more clinically relevant?
Right. So the challenges of multimodal data are that these databases in general came about before machine learning, and they're not optimized for machine learning. So for every data modality, you have to spend a fair amount of time just hands-on understanding what's going on a conceptual level, but also engineering it to be performant in your pipeline so that your GPUs aren't waiting to load data from disk, which is very wasteful. So the second part of that question is how to make protein engineering more clinically relevant. Once again, the caveat here is that I don't really work on clinical things. I work on the far upstream molecular things. But there are a rather astounding number of companies, big and small now, that are using protein in machine learning and protein engineering to design therapeutics. There's a lot of focus on antibody design, just because antibodies are somewhat relatively easy to design, and they're proven as therapeutics. But there's also really creative things people do, like, can we take a biologic and make it less immunoreactive so that your immune system doesn't reject it, or things like that. So, or even like, can we design better gene editors? So there's a whole ecosystem now. Like, it's an astounding number of companies doing this.
Okay. Thank you for the answer, sir. This was our last question. On behalf of myself and all our participants, I would like to thank you for spending your valuable time transferring your knowledge to us and for this informative presentation.
Thanks for having me.
I wish you healthy days.
You too. Have a great day.
Thank you.